So the learning outcome for today's lesson is to outline how microarrays are used in the analysis of genomes. Genomes means all the DNA and all the genes present in the nucleus or a cell or an organism. And in detecting mRNA in the studies of gene expression. So now we're going to look at the powerful tool of genetic technology, and this tool is called microarray. So what is microarray? Microarray is a small piece of glass or plastic, and its size is usually about 2 mm times 2 mm, 2 cm times 2 cm only. Not 2 cm cube, but it's square. So it's actually square here, cube. Okay, so this is cube. Okay, so like this picture here, right? And within this piece of glass or plastic, it contains approximately 10,000 or more spots in rows and columns. So if you see this one here, you can see a lot of tiny spots, right? And these spots are arranged in rows and in columns. So you got 10,000 spots in rows and columns per cm square. So let's say, for example, here is 1 cm square, right? So this 1 cm square. So this 1 cm square itself, okay, I don't draw it, not so accurate. Yeah? Let's say this 1 cm square. Let me draw again. So let's say this 1 cm square, okay, I need to draw like this, contain 10,000 number of spots in rows and columns. And each spot, so we enlarge the spot, each spot here, each spot contains multiple identical strands of DNA probes. So you can see here, this is one spot, and in one spot in this picture, it shows 4 identical DNA probe. So you remember, DNA probe is a single-stranded DNA where the bases can be complementary to the bases of the DNA that we want to find or we want to fish out. Okay, so the concentration of the DNA in one spot is in picomoles. Picomoles means 10 to the power negative 12 moles. So these DNA probes are attached to this solid surface, which is glass or plastic. So these probes are used to identify DNA or RNA, which are having complementary base sequence with it. So this is the microarray. So remember, I'm going to recap again what you have learned about the DNA probe. So this single strand of DNA is in yellow color is the DNA probe. So DNA probe is a short single-stranded DNA, usually about 100 to 1,000 bases long. It has got base sequences that is complementary to the DNA that we wish to find or identify. So like this case here, it's complementary to this particular strand of DNA that we want to find. So therefore, our DNA probe will be able to bind to a specific gene or allele that we try to find from the genome of a cell, for example. Or we are trying to locate VNTR or STR region, which is used in genetic fingerprinting. So this is the use of genetic probe. So this microarray here is very interestingly, is also known as DNA chip or gene chip because it's very small, but in this very tiny little object, it contains many information. In this case here, many DNA probes. Sure. Okay, so just like our, your ATM card or your IC, right? You got a chip that is gold in color. So this chip actually stores a lot of information. So in a tiny little object, it contains many information that's stored in it. So that's why it's called the chip. So this one has got what information? The DNA, which is the DNA probe. So they call it DNA chip or gene chip. So if the probe is able to bind to the gene, so we call it the gene chip. So this microarray can be made by automated process. That means using machine. So we can tell the machine, okay, I want to make a microarray and I want these DNA probe to be included in the tiny little spots in the microarray. So every spot got different gene probe or DNA probe. So I can design it and instruct the machine to make this microarray. So a huge range of genes can be searched from the databases of human genome. So you know that scientists have involved in many years in the Human Genome Project. Have you heard about the Human Genome Project? So this is a project that's taken by many scientists in the world for many years in order to sequence all the DNA of human. So they try to find out the base sequences of all the DNA of a human. And from there, they try to find out which sequence are the gene. So because of that, they also found out base sequences of the gene. So this is a very huge project that is now completed already. So therefore, all this information about the DNA base sequence of our human DNA has been available and stored in databases. Databases means data means the result of the research, right? Bases means is a place where we store them. It's like library. So the scientists can go to the databases of human genome to look for the base sequences of the gene in order to make the DNA probe. So I want to find out whether this person has got this, let's say for example, I want to know whether this person is a carrier for hemophilia. So carrier for hemophilia means you got two alleles, right? One allele is normal and one allele is for hemophilia. So I want to go to the database and look for the DNA base sequences for hemophilia allele, and then make it into DNA probe and put it into this microarray, and then use this to find out whether any other people, to find out from the sample, let's say blood sample of a person, whether this person is a carrier for hemophilia. So I want to know whether this person got this allele or not. So I can create this DNA probe and then find out whether this person got this allele. So it's very interesting, isn't it? How this is being done. So my question here is, now this is a microarray, very small, 2 cm times 2 cm. So my question is it possible to attach all the 21,000 genes from the human genome into one DNA chip? Yes or no, and explain why. So just now we saw from the information of the definition of microarray, right? We say that every 1 cm cube can contain at least, minimum, right? 10,000 spots, you know, and each spot can contain a different DNA probe. So how big should be the area required to contain 21,000 of genes? Yes. So we need 2 cm cube, right? Need minimum slightly more than actually 2 cm cube, a square, to contain 20,000 spots, right? And slightly more than that. So which not 2 cm cube, actually not 2 cm cube. 2 cm cube will contain 40,000 spots already. Can you all see? So if this is 2 cm cube, so each of these smallest box here is 1 cm cube, right? So 1 cm cube can contain 10,000 spots, you know. So if it's 2 cm cube, isn't it? I can contain 40,000 spots. 1 cm cube can contain 10,000 spots. 2 cm cube, 40,000 spots, which is actually more than enough for us to put all the 21,000 gene probe into this one microarray. Right? Wonderful. Huh? So that means one microchip, one gene chip is enough to contain all the DNA probe, the gene probe that can identify all the genes of the human. It's amazing, right? Okay, so before we proceed, let us recap first what you have learned. So you have learned the probe, and you also learned about the primer. They are very similar because both of them are single-stranded, short single-stranded DNA, but their function are different. So explain the difference between the probe and the primer. Can you remember? So to make it quick, so I'll just show the answer. So the probe is a short length of single-stranded DNA that attaches to one end of another single-stranded DNA. So this is a probe here. So short length of single-stranded DNA which attach to another single-stranded DNA at one end. And the probe is usually labeled, so in this case here, with radioisotope, or it can be labeled with fluorescent stain. And the position of the probe will indicate the position of the DNA. So this is when you learn about the electrophoresis. So after electrophoresis, the DNA fragments are actually located in different locations. So out of the all the total bands there, you want to know which band contain insulin gene. So you have to use the probe to find out, or maybe this one contain insulin gene, right? So this one here, contain insulin gene. So you use the probe to find out the location of the gene among the bands that are formed after electrophoresis. So it is used to locate the position of the DNA. Now, what about primer? So usually primer is needed for during DNA replication. So how do you define primer? So primer again is also a short length of single-stranded DNA that will attach to one end of another single-stranded DNA. So its purpose or its function is to allow DNA polymerase to bind to it, to start to make a complementary strand using this single template strand. So it's used during DNA replication. So you need to know what is a probe and what is a primer and what are their functions which are different from each other. So now we have learned what is microarray. We want to see, so what the scientists have used the microarrays for. Scientists have used the DNA microarrays for, number one, comparative genomic hybridization, and number two, gene expression profiling. So what do these two things mean? Comparative genomic hybridization means we have used microarray to compare the genes between different species, different individual, or different cell. Hybridization, it means by joining them together with the DNA probe in the microarray. So comparative genomic hybridization means to compare the genes that are present in two different species or individual or cells. So that is each gene. So let's say, for example, I want to see the insulin gene. So I want to know that whether insulin gene is present in both the species. So none of the species got the gene, or between the two species, one of them have, one of them don't have, or both of them have the gene. So to see whether the organism contain the gene or not. So this is very useful to find out genetic relatedness of the two species. So if they have the genes, that means they are highly related to each other. If one got, one don't have, that means they are not so related to each other. So the more closely our gene resemble one another, the more closely we are related to each other. That's why brother and sister look more alike, relatives look more alike than non-relative, because they will have got more genes that are similar to one another. So it shows the genetic relatedness between individuals as well as between different species. For example, human and monkey and chimpanzee, they are more closely related compared to human and maybe a cockroach, correct or not? Okay. So there'll be more genes that are very similar between us and the chimpanzee versus between us and the cockroach. Okay. This first use of DNA microarray. Second use is for gene expression profiling. That means we want to find out the expression levels of thousands of genes simultaneously at any specific time in each cell. That means we want to know whether in a particular cell, a particular gene is being expressed or not. Yes or no, and then the degree of expression. That means, does it cause a lot of insulin to be produced or a little bit insulin to be produced at different timing? So when glucose is present, high level of insulin produced. When glucose is absent, no insulin produced. So do you want to monitor the gene expression? So we want to identify the genes that are being transcribed into mRNA, because when the gene is expressed, it is actually transcribed into mRNA. So we want to find out what are the genes that have been transcribed into mRNA, which cell will transcribe it, at what time it will transcribe it, to what level it will transcribe it. So this will help us to study the effects of diseases. For example, a normal cell versus a cancer cell. What's the difference? What is the gene in a cancer cell that's being switched on that normal cell will not switch on? Okay, then so this is one example lah. So what kind of treatments I've given to the cell? So when I give the drug to the cell, how does the cell respond? What is the gene that's being switched on when I give this treatment? When the treatment is effective, what genes are switched on? When the treatment is not effective, what genes are switched on that's not effective? I can also use it to study development stages. That means, you know, when the zygote starts to grow into embryo and embryo into fetus, at different timing of development, let's say 9 months of gestation, 9 months of pregnancy, at different months, what organs are being developed? So in order to develop that organ, what genes are being switched on to cause that organ to be developed? So we can use gene expression to actually find out in sequence during development what genes is switched on at what time, and then switch off at what time, which other genes being switched on and switch off, and so on. So it's very wonderful tool. So therefore, we're going to look at these two examples and the procedure of carrying out them. Now, this is the notes for you. I'm not going to read the notes. You read it yourself. I'm going to use a diagram to explain it, the procedure. So the first use of the microarray is to find out the absent and present of certain gene in 2 species. So in this example, I'm going to see how we use microarray to find out the absent and present of certain gene in these 2 species. So therefore, I have to obtain the DNA from these 2 species. So I collect the DNA, that means the genome. I collect the genome from these 2 species, and then I cut those DNA with restriction enzyme so that many fragments of different length are formed. And then I denature the DNA to make all this DNA single-stranded. So after that, I will label this DNA. So from species A, I label it with a red color fluorescent tag. For species B DNA, I label it with green color fluorescent tag. After labeling, I will mix this DNA onto the microarray. And my microarray already got a lot of DNA probes that already fixed onto it or immobilized onto it. Means you stick those DNA probes into the surface of this glass. So therefore, the DNA from species A and species B, they will actually bind to the DNA probe if it's complementary basis, if they are complementary. So allow time for binding to happen, and then after that, we wash off the unbound single-stranded DNA, and then we observe the spots using UV lights, because those tags are fluorescent. So when we shine the microarray with UV light, whichever part which contain those DNA with fluorescent tag, it will fluoresce. Okay, so this picture here is example of a result. So you can see a lot of spots, and each spot got different color. So how to read the color? Okay, so you see that now the spot can be red color or green color or yellow color. Okay, so you see, if this is species 1 and species 2, so this is species A, and this is species B's DNA. Species B's DNA. So species A DNA are labeled with red fluorescent tag, and species B DNA labeled with green fluorescent tag. So case number 1 is species A contain the DNA and species B don't have. In that particular spot, it will give us the red color. So in this example here, you see, when a spot is red color, it shows that it has got DNA from sample A, it doesn't have DNA from sample B. When a spot is green color, meaning it's got DNA from sample A, DNA from sample B. When a spot is green color, it's got DNA, it doesn't have DNA from sample A, it's got DNA from sample B. So by looking at the color of the spot, it will immediately tell us whether species A and species B contain the gene or not, so present or absent. So this one is present in only A, this gene is only present in B, and this gene is present in both species A and species B. So after we analyze the result, we can do a laser scanning to scan the microarray and then analyze and store the data in digital form so that we can refer to it in the future. So this is a homework. You can fill in the blank on your own, like what I illustrated to you just now. How to analyze the data. So just now we look at how to use the microarray to find out the presence and absence of gene in two species. Now we want to look at the second use of the microarray is for gene expression profiling. So this part here is the notes for you to read on your own. You can fill in the blank, it's very easy. I'm going to use this diagram to explain the procedure. So here we're going to compare between cancer cell and a normal cell. What are the genes that are being expressed in cancer cell that is not expressed in normal cell? So number one, we will try to isolate RNA from these two cells, because when a gene is being expressed, the gene will be transcribed into mRNA. So we want to isolate the mRNA. So after isolating the mRNA, now because we are using DNA probe, DNA cannot bind to RNA. So DNA must, the RNA must be converted to DNA so it can bind to DNA probe. So the next step is to convert the mRNA to DNA by reverse transcription. So after reverse transcription, we get our cDNA, the double-stranded cDNA, means the complementary DNA. We got a very small copy of it, so we have to amplify it with PCR technique. So PCR technique is to amplify the double-stranded cDNA. So we make many copies of them. So after that, we will label this cDNA, and then we denature the cDNA so it becomes single-stranded. Denature means we break the hydrogen bonds between the complementary bases of the two strands. Right? We break those bonds so that they become single-stranded. Okay. So after that, we will mix it with the microarray so that this single-stranded DNA can hybridize with the probes that are complementary to it in the microarray. So if it's able to hybridize, then that spot will fluoresce, telling us the presence of this mRNA. That means the gene is actually expressed in this cancer cell. So this is how we use to detect whether a certain gene is being expressed, and also it depends on the intensity of the spot. Intensity means brightness of the spot. So the brighter it is, that means to say that brighter means the more you have the cDNA. That means to say that the more we have the mRNA, that means to say that the more is being expressed. So therefore, we can know whether number one, the gene is expressed or not, and number two, the degree of the gene expression. Means higher or lower, more or less. So these are the two information we can get from this procedure. So there's this experiment that you can actually do it in this link here, in showing you how to use microarrays to compare the differences in gene expression levels between cancer cells and healthy cells. So if you have time, you're interested, you can actually click and play this one at your own free time, not now, okay? Not now. So in this topic, you have learned the definition of microarray, the uses of the microarray. I will only introduce 2 uses to you, okay? And to outline how microarray are used in the comparative genomic analysis, means you want to know presence and absence of the gene in an organism and compare between 2 organisms also can. Gene expression profiling means whether a gene is being expressed and the degree of the gene being expressed. So that's all for this part for microarray.